Good morning, and thank you for the introduction. So today I'll show you how long read has revealed a common but yet so far hidden cause of SORD-CMT, namely a gene-pseudogene inversion. So pseudogenes are copies of coding genes that were once considered non-functional but are now recognized as relevant to human disease. And they can arise from two main mechanisms: either by retrotranscription of mRNA with loss of introns and regulatory elements, and these are processed pseudogenes, or by duplication of the parent gene followed by accumulation of inactivating mutations. These are unprocessed pseudogenes that maintain an exon intron structure and a high similarity to the parent gene. It is this high homology that makes them more prone to recombination with the parent gene, leading to potentially pathogenic events. Those are gene conversions where short segments are transferred from the pseudogene to the gene, unequal crossing over leading to duplication or deletion of the gene, and finally inversions where segments of DNA are exchanged between the pseudogene and the gene without a copy number variation. And while there are many examples of the first two cases, only two gene-pseudogenes inversions are known in IDS and F8, associated respectively with Hunter syndrome and hemophilia A. So this poses a question: are they really so rare, or are they simply being missed? In fact, this high homology combined with the absence of copy number variation makes it very difficult to detect the inversions. In fact, in short read, even reads that span the breakpoints, like the one in red here, normally contain at most one SNP that is not sufficient to tell the inversion apart from the reference. By contrast, with long read, we have a better chance to span multiple SNPs across the breakpoints, and even if the reads are still not long enough to capture the entire inverted fragments, these mismatches can still signal the breakpoints of the inversions. So with that in mind, our aim was to close this gap in knowledge and to use long read to characterize these gene-pseudogenes inversions, first by assessing their presence in healthy individuals using a public database of long read from the 1K Genome Project, and secondly, to investigate the relevance in unsolved rare diseases, of course, particularly in inherited neuropathies. So we started by selecting from the over 10,000 pseudogenes in the Scicube catalog only the gene-pseudogene pairs that had a configuration permissive for inversion. So specifically, the pseudogene had to be unprocessed and to lie on the same chromosome with the parent gene, but in opposite orientation. And in this way, we came to a list of around 400 candidate gene-pseudogene pairs. And interesting, almost 20% of the parent genes were present in the HOMIN database, so meaning they were already associated with human disease. So checking for the actual occurrence of inversions of the 1K Genome Project population, we identified a total of 37 inversion events in 9 gene-pseudogene pairs, meaning that almost 4% of the genomes contained one inversion. And two genes stood out as clinically relevant. One, of course, is SORD, which is a common cause of CMT, and also the locus with the highest number of inversions, and the other being PMS2, which is related to an hereditary cancer syndrome. Although in both cases, all these inversions were predicted to be neutral. Importantly, only one inversion was already reported in NOMAD, meaning that these inversions are indeed mostly invisible to short read. So as said, SORD was particularly relevant for us, and interesting, even if now biallelic mutations in this gene are known as a common cause of CMT SORD, their discovery was delayed for years precisely because of the presence of the pseudogene. In fact, short read struggles to distinguish between the two, and one of the most common mutation in SORD is actually fixed in SORD2P. I want to stress once again that, and you will see why, that SORD neuropathy has a reliable disease biomarker because the disruption of sorbitol dehydrogenase leads to the accumulation of sorbitol in tissues. So searching for SORD cases in our CMT cohort, we found 3 patients with high sorbitol levels, but only 1 pathogenic variant in SORD according to short read. So a second pathogenic variant was likely there, and based on our finding, we hypothesized that this could be a SORD2P inversion. So to test this hypothesis, we performed long-read sequencing and optical genome mapping in all 3 cases. And we indeed found a recurrent inversion in all of them, this time pathogenic due to the breakpoint location that disrupt the SORD reading frame. And here you can see the long-read and short-read data in both patients and a control. And in long-read, you have, as pointed by the red arrows, these split reads, so reads that are SORD reads are partially mapped to SORD2P and vice versa, that mark the inversion breakpoints and are absent in the control. By contrast, nothing is visible in short-read, and the patient looks identical to the control. We confirmed the presence of the inversion with optical genome mapping, which is highly suitable for these large structural variants. And here you can see the inverted interval very clear, and the breakpoint coincide with those detected with long read. We screened for, so using a PCR method with breakpoint-specific primers, we screened for additional CMT cases. And here you can see an example of the 3 kilobases product that is absent in control. So we identified the inversion almost a quarter of the CMT patient that had only 1 pathogenic variant in SORD, but this number came, like the percentage came to up to more than 60% in those that also had a high sorbitol. So the screening for the inversion is now highly suggestive for this kind of patient. And for the remaining high sorbitol patients, long-read proved once again to be very useful, as we were able to solve them all. 1 patient surprisingly carried a different pathogenic inversion with distinct breakpoints, thus the negativity of the PCR. And the other 2 patients carried 2-point mutations that were missed by short read because of ambiguous mapping. In the end, these inversions that once again are completely invisible to short read are one of the most common pathogenic allele in CMT SORD. And this finding had direct clinical implications for the patients, as 2 of them were enrolled in the clinical trial we heard about yesterday on the efficacy of govorestat. Finally, we ask ourselves, why are these inversions so common? And the answer may lie in the 3D chromatin structure. So we looked at the micro-C tracks that are heat map, where the brighter the red color is, and the more strong and frequent are the interactions between 2 loci. And so you can see by this striking interaction pattern between SORD2P and SORD, they seem to be in high physical proximity, probably because of a chromatin loop, and it maybe is this physical proximity that promotes the occurrence of the inversion. And this appears to be a general trend in our candidate gene pseudogene pairs. We performed the FIT-IC analysis on, to study the chromatin contact frequency in our gene pseudogene pair, comparing them with those in random selectively gene pairs matched by distance, but that were non-paralog. And we found indeed a significant enrichment, suggesting that maybe is the homology that promotes the physical proximity that in turn drives the recurrence of inversions. So to conclude, I demonstrated for the first time that these gene pseudogenes inversions that are actually mostly invisible to short read are actually relatively common in the human genome. Most importantly, we demonstrated that one of them, the SORD2P inversion, is actually one of the most common pathogenic allele in one of the most common forms of CMT. And this was a clinically actionable findings because patients were enrolled in clinical trials. We also speculated that it's the chromatin contact between genes and pseudogenes that that has a factor that facilitates the occurrence of inversions. Finally, it's easy to speculate that inversions in other gene pseudogenes are part of the missing irritability, a part that can now be explored thanks to the new sequencing technologies. So, of course, I will end by thanking all the people and the funders that contributed to this discovery, particularly the CMTA and the FM Telethon. Thank you. Thank you.
Thank you to Dr. Quartesan. Any questions? Yes, Dr. Rari.
Thank you very much. That's a very interesting story. I mean, I think it's really interesting, the hiddenness of this. So it's not only that the inversion, as now the second commonest SORD variant, was hidden, but the original common one was hidden.
Exactly.
So people that have cohorts of patients that haven't done it recently, they may be missing both. So they won't even think of doing the sorbitol because they have one variant. Yeah. So, you know, it's not, so I think what people may not realise, it depends on how they get their samples done or how they get their testing done, is they may miss the first variant. Yeah. So, so that's, I mean, it's interesting. So that's a huge possibility for other genes. Have you explored any other genes yet?
We had explored, because one of the genes was SVIL, which is related to an inherited myopathy, and we looked at the genomic singlet data, but really not seeing a candidate, but we are exploring the possibilities, the actual possibility of other pseudogenes being relevant.
Yes?
A lovely talk. I'm curious, in the States, sometimes we get a clue from being able to order the urine tests. And is that possible in Europe? I'm just ignorant of that. Urine, yes.
So we usually do it in blood, not urine. But I think, do you do it in people without, one of the questions is, should sorbitol be done in urine or blood in CMT without a variant?
Well, if we have a pure motor neuropathy, often, or predominantly motor neuropathy, we frequently will do that. It's just a quick screen.
Yeah. I mean, that's a question from this meeting, because, you know, if people, I think that's a very good question, which maybe we can address, is because not everyone will have even the way of picking up the common mutation. But I'm not sure how widely available sorbitol levels are around the world, rather than just...
Yes, I'm asking.
You know, I don't know. Catherine's going to have a comment there?
Is the Mayo Clinic urine sorbitol test is available?
That's in the US.
No, but they have a global operation across 38 countries where you can send it in.
Yeah? Yeah, we do have a question there.
Thank you, very interesting. My question is related to the chromatin.
Yeah.
Which hypothesis do you have to explain how chromatin is working to facilitate this crossover between both the gene and the pseudogene? I mean, do you have, for instance, data from episignature in these patients or something like that that could explain changes in chromatin?
Well, we actually don't have them. Like, generally speaking, this was more like an exploratory study, you know, on a genome-wide level, but this is indeed a good suggestion, so maybe we should really more look into it and more specifically look in which are the actual factors that promote these higher chromatin interactions, yeah.
Thank you.
I think we have another question in the back somewhere, right?
Yeah,
Okay.
all the way back. I had a question about the list of genes that you showed that actually have similar inversions, because quite a lot of them have, and also the gene, the SORD, have really large introns, and they usually also have a specialized form of pre-RNA splicing. Yeah. Have you maybe checked if this mechanism itself could lead to the increased inversions?
So we haven't checked that, but that's actually a very good suggestion. Thank you.